So you've run either your reads or your contigs through the comprehensive genome analysis service in Patrick. Now, after you've got that, your genome is part of the Patrick system. It's only available for you to view, but you can compare it to any of the public genomes in Patrick or any of the private genomes that you have. So how do you find that genome so that we can show you what it looks like? Well, you could go down here and click on your jobs monitor. You could search for it in the global search, or you could go up into workspaces and click on My Genomes or My Jobs. I annotate a lot of genomes and I can't often remember exactly what they're called because I use like SRR numbers and I can't remember all of that. So I often like to go through My Jobs, but you know, if you gave it a distinctive name, you might find it more easily that way. So when I want to view something, you highlight the row, and this populates the vertical green bar with possible downstream functions. So I want to view this job. So I click on View. This takes me to the job landing page, which I've discussed many times in other videos, and you're probably bored to hear about it. But one thing I never discussed was this interesting little blue eyeball up here in the upper right corner staring at you that I haven't clicked on yet. So this will give me direct access to my genome page. And so let's click on that, View Annotated Genome. It's going to overwrite this page and show you all the information associated with your particular genome in Patrick. Let me point out that there's a lot of stuff that comes with the genome annotation in Patrick. We can't fit it all on one page. So we have a number of tabs here so that you can see all of those different things. Another thing that I should note is that here you can share the genome. If you're working with a group, you want to share this genome with other people, you could click here on the share icon and you could start adding lab members or whoever you want to share the data with. And if you don't know, you just know their last name, you can put a name in. Oh yeah, this is the guy I want. And I can give him view or edit privileges depending upon, you know, if you're teaching a class, you might just want it to be view. If it's a collaborator, you might want it to be edit. So you can say add user and save that. And then in a while, you'll be able to see, see, and it shows who has the privileges to view this gene. So let's talk genome. So let's talk a little bit about this page. With any annotation in Patrick, it comes, here's the name, it tells you where your genome is within the great tree of bacterial life, the taxonomy up here in the breadcrumb. There's information about the genome itself, its quality, how many contigs it has when it was inserted into the system. There's also details about the genomic features, the number of genes called, repeat regions, tRNA and rRNA. Notice that there's Patrick annotation, but no RefSeq annotation because this is a private genome. Protein features, getting into the specifics on that, how many hypothetical proteins, proteins with functional assignments, all the information here. And these are all hyperlinks, so you could go directly to that information. And then specialty genes. Any genome annotated in Patrick is blasted or blatted against specific databases for virulence factors, antimicrobial resistance genes, drug targets, transporters, or human homologs. So we have that kind of information for you that you can drill down into. You'll also notice that we have a number of tabs here that show different things about the genomes, including if there are antimicrobial resistance phenotypes, the phylogeny, not necessarily, your organism won't be included in it, but it shows you an overall phylogeny of either the genus or the order. There's a Genome Browser, a circular view. Sequences are the contigs themselves, and it tells you how big they are and how many genes are called on them. features are a list of all the genes. The specialty genes, if we were to click on that, just because this is kind of interesting for people, it shows you whether they were a blast search or a K-mer search, which we do for some of the antimicrobial resistance. So I could click here to zero in on that in filters. And if I wanted to see everything, you can see that they're the blast ones, which show you the E-value and the identity, and also links to publications that describe this gene as, in this case, a transporter, which I can see from this top row here, or as in this case, this one is a virulence factor. And I could get to the direct publication here. And look, you can see here, there's some information about this that appears when you select one at a time, that tells you the information about those things in that lower, that are in that selected gene. So this is a valuable thing, but if you wanted to see everything that was identified by K-mer search compared to that, you could click here and that would show you what the K-mer searches were and what they were hitting within this particular genome. Protein families, we have special tabs for that. For pathways and subsystems. Now, subsystems are a special annotation project of grouping genes together that function together, like maybe they're pathways, maybe they're part of a transporter system. We have a curation team that dig into these kind of things. So this might be something that you want to explore as well. Transcriptomics, we won't have any for your private genomes, but for some of our more that we have a lot of data for, like Mycobacterium tuberculosis, you could see gene expression data from some of the public experiments and interactions. So this is what your genome looks like in Patrick, and once it's here and you have all this information at your fingertips, you can do a lot of things with this. You can build trees including your genomes. You can compare the protein families. You can use it as a reference genome and compare SNPs of other, using reads from, if you're doing an experiment with it. But the first step is you have to get it to this point and have to get it inserted into the Patrick system. So if you have any questions, you need to let us know, but we hope that you use our service inside us and have fun exploring your genome in the Patrick tools. Thank you.